This video is an explanation of the reading comprehension passage about clinical trials. This could be watched in conjunction with any question about the passage, and of course each question is explained in its own video explanation. I will say if you're doing multiple questions about this passage, I would encourage you to do all the questions first, then watch this video, and that way you'll answer all the questions with your own understanding of the passage. So here's the passage. Paragraph 1. The efficacy of standard clinical trials in medicine has recently become the subject of contentious debate between those practitioners who maintain that such trials, despite admitted shortcomings, still represent the best means we have for learning about the effects of pharmaceutical drugs on the human body, and those who maintain that the current system of collecting knowledge of such effects is but one possibility and most likely not the most efficacious one. Gimli and Lebsmith, in their recent work, fall into the latter camp and indeed go further by challenging the idea that standard medical trials can yield meaningful information on pharmacogenetics, or how a drug interacts with the human body. So let's stop right there. What's the nature of this debate? And so there are standard clinical trials, and presumably we'll find out a little more about what is meant by standard clinical trials, but there are some people that are saying standard clinical trials are the best way to go. They're not saying that they're perfect, there are shortcomings, but this is the best way to go. And then there are other people that are saying, no, there are better systems. And in particular, Gimli and Lebsmith seem to be really criticizing the status quo and saying that the standard medical trials are deeply flawed, and they don't yield meaningful information on how drugs interact with the human body. Gimli and Lebsmith's foremost criticism is that the effects of a drug differ depending on a person's physiology. To be sure, there are cohorts or groups that react to a drug in a specific manner, but clinical trials are unequipped to identify such groupings. The main reason is that clinical trials are allied to the notion that the larger the number of subjects in the study, the greater the validity of a drug, should it show any promise. Therefore, even if a drug can exercise a marked effect on a subset of subjects within a trial, this information will be lost in the statistical noise. So that's a really complicated paragraph. What's going on there? So we find out there that the standard clinical trials, the status quo, the standard clinical trials just use a large number of people. Say they use 1,000 or 2,000 people. And the idea is, well, if the drug works for 2,000 people, it must really work. What Gimli and Lebsmith are saying, suppose it's just one portion of the population that has this marked effect to the drug. Just some subgroup of people that have a particular genetic difference, and it may be, allied to anything, height or freckles or hair color or eye color, anything like that. But for whatever reason, there's this one particular subgroup, and the medicine really works for them, and it doesn't necessarily work all that effectively for anyone else. Well, of course, you wouldn't see that because there'd be a small mix of those people among the 2,000 people tested, and those 20 or 30 people would show a marked increase, but that would just be lost in the randomness. Some people would show an increase, some people wouldn't, and it would look like the drug was not particularly effective. And so the clinical trials are not designed to pick out, is it going to work for a particular subgroup? There's no way that the subgroups would be isolated by the current statistical procedures. Third paragraph. Another criticism of Gimli and Lebsmith concerns the very idea of validity. Pharmaceutical companies will run hundreds of trials on different medications. Given the sheer number of trials, a few are likely to yield positive results even if there's no demonstrable effect. Gimli and Lebsmith cite the fact that most pharmaceuticals have exerted a positive effect in the first round of testing are likely to fail in the second round of testing. So this is another really sophisticated statistical idea. The basic idea here is that something is statistically significant if you see results that are highly improbable. And suppose that the results are only going to happen, suppose we set a bar and we say if something happens only 1 out of 100 times, then we'll call it statistically significant, okay? If it's a 1 out of 100 chance of happening, that's statistically significant. Well, of course, if I run 1,000 different tests on 1,000 different chemicals, about 1% of them will have results that show up 1% of the time, 1 out of 100 times, just purely by chance. if you run a large number of trials, you'll get the things that are rare once in a while, the things that just arise purely by chance. And in the first round, those are identified as statistically significant. People saying, oh, wow, this is something that really works. It has a result that you'd only see one time out of 100. But then, of course, when they try it again, they don't get that because it was purely a chance result. That's really a very, very subtle idea in that paragraph. Gimli and Lebsmith argue that a more effective approach is to identify groups who exhibit similar genetic subtypes. This very approach is currently in use in groups possessing a particular molecular subtype of breast cancer. Furthermore, these groups are not only trying one specific drug, but also a combination of such drugs, subbing them in and out to measure the different effects on a subject, a procedure that Gimli and Lebsmith endorse. Nonetheless, such an approach is often time-consuming and costly. However, given the constraints of current medical trials, trials that target subtypes, even if they do not yield any significant advances, will encourage a culture of experimentation on how clinical trials are conducted in the first place. So that's the end of the passage. Notice a few things. Notice that the author of the passage appears to be a real fan of the work of Gimli and Lebsmith. They're doing a lot of describing of Gimli and Lebsmith, but they appear to be showing them in a very good light. They appear to be suggesting all their criticisms are really good criticisms and all their ideas are really good ideas. And so we can infer that the author really supports their analysis. And so the main idea is certainly about showing Gimli and Lebsmith analysis, their criticisms, in the way that standard medical trials are often conducted. Now, what they're advocating is doing something where they're identifying groups that have similar genetic subtypes. We're not told how you would identify those people, but some of them identify them and then test those individual groups. They point out that this is done already for one particular kind of breast cancer. Now, the problem is that this is time-consuming and expensive, but because they've identified all these obvious shortcomings of the traditional approach, just doing this kind of research, now it may lead to wild success, but even if it doesn't, it would encourage experimentation. It would encourage people to rethink the idea that we do medical trials in the first place.